Hello friends, today we are going to discuss about life cycle and pathogenicity of Entamoeba histolytica. Introduction. Entamoeba histolytica is a pathogenic amoeba that is widely known to cause intestinal and extraintestinal infections in humans. Entamoeba histolytica belongs to the class Protozoa, subclass Sarcodina, and order Lobosa. Entamoeba histolytica causes dysentery by invading the mucosa and submucosa of the large intestine in humans. So, this histolytica is commonly found in tropical and subtropical countries. The life cycle of Entamoeba histolytica begins and ends within a single host, that is a human individual. Entamoeba histolytica life cycle. Cyst stage 1. Infection occurs from injection of mature cyst in water or food contaminated with feces. Due to the protection of the walls, the cyst persists for several days and sometimes weeks. They are responsible for transmission. Excitation stage 2. Excitation is the process by which follicles transform into vegetative cells. When the cyst enters the ileum of the host small intestine, exocytosis begins. Trophozoites are released in the small intestine and from there they migrate to the large intestine. Trophozoite stage 3. Trophozoites are single-celled parasites 14 to 18 mm in diameter. They multiply in the small intestine by binary fission to produce cysts that are excreted in human feces. Several trophozoites remain inside the lumen of the small intestine. The rest attach themselves of the intestinal mucosa that enter the bloodstream and further grow in the extraintestinal regions of the host like lungs, liver and brain. Conclusion. Entamoeba histolytica invasion in human host vary from showing zero symptoms to being fatal. This parasite, discovered in 1859 by Lambel S. Jardine, has shown the distinction between pathogenic and non-pathogenic amoeba species. Pathogenicity of Entamoeba histolytica. The pathogenesis of Entamoeba histolytica is discussed from an immunological point of view. Evidence that some trigger mechanisms transform communal living organisms into tissue invasive pathogens is rejected as insufficient. The number of cases of liver abscesses compared to the number of intestinal amoebic infection in the population is so low that this in itself indicates that the tissue invasion is a rare event in the life history of amoeba. Experimental evidence has been reviewed that requires some kind of sensation before an amoeba can invade tissue. When postulate the immunologic basis for amoeba pathogenesis, a parallel is created between the behavior of malignant cells in the body and amoebic infection in the gut. One compelling hypothesis that deserves further research effort is that the altered immune response is the basis of the pathogenic mechanisms in the host. So that's all about your life cycle and pathogenicity of Entamoeba histolytica. Thank you.